The overall goal of this procedure is to label proteins that reside at the cell surface with a cell membrane impermeable form of biotin containing a disulfide bond in order to quantify protein trafficking through the endocytic and recycling compartment. This is accomplished by first labeling lysine residues in the extracellular domains of plasma membrane proteins at 4 degrees Celsius. The second step is to induce endocytosis of biotinylated plasma membrane proteins by warming the cells to 37 degrees Celsius for specific periods of time. Next, cells are cooled again to 4 degrees Celsius to stop intracellular trafficking, and the biotin is cleaved off with a cell membrane impermeable reducing agent so that the proteins that remain labeled are only those that were endocytosed and thus are protected from the reducing agent. The final step is to lyse the cells and incubate the lysates with streptavidin beads to isolate the biotinylated proteins. Ultimately, the biotinylated proteins are eluted from beads and separated on a protein gel, and the protein of interest is detected by immunoblotting followed by densitometry to quantify its endocytosis and recycling.
This technique can be used to investigate diseases that result from a trafficking defect of cell surface proteins, such as transporters, channels, and receptors.
Generally, the reason for the assay not giving consistent results is the lack of strict temperature control.
To biotinylate the cell surface proteins, culture the cells on 6 24mm filters. Transfer the filters quickly to cold PBS on ice, allowing the buffer to quickly cover